Thank you very much for the introduction. Good morning to all of you. So we developed a method which is alignment free to predict human SS3 domain-peptide interaction using graph kernel. So SS3 domains are an important class of peptide recognition module and perhaps most widespread protein domain found in protein database. The domain specifically recognize short linear proline-rich peptide and involved in many cellular processes such as signal transduction, cell communication, growth, and differentiation. So SS3 domains are basically 60 amino acid in length, and it has a conserved beta barrel structure consisted with 5 to 6 beta strand. So around 300 of SS3 domains are known in human proteins. Interestingly, 25% of human proteins contain proline-rich regions. So these regions it can be targeted by the SS3 domains. So here is the challenge is to understand how the hundreds of SS3 domains specifically bind with their partners to regulate specific biological functions. So there are many motifs have been reported previously that can bind with SS3 domain. They are normally categorized into two, canonical motif, peptide motif, and non-canonical or atypical peptide motif. The length of the peptide is vary from 5 to 15 amino acids. So when you talk about the canonical peptide motif, it can be further classified into two types, class 1 and class 2. Both type of motif contain one core in red, PX5P. So here X is any kind of amino acid and phi is hydrophobic amino acid. But in class 1 peptide, there is a basic amino acid upstream of the sequences, upstream of the core motif. So the basic residue is normally arginine or lysine. And for class 2, the basic residue present in the downstream of the sequences. So the class 1 motif and class 2 motif is similar with a different orientation. And there are lots of non-canonical motifs. I'm giving you some example, but there are many more. So till date, there are several approaches have been reported to predict SS3 peptide interactions. They are mainly based on PWM approach. So there is one tool, which is state-of-the-art, multiple specificity identifier. So there are some drawbacks of multiple, PWM approaches, as you have seen in last talk from last speaker. So they are linear model. That means they do not consider the correlation between the amino acid position. And they also do not model the non-interaction. So these PWM approaches is only based on the positive interaction. So they ignore the negative interactions. And the method based on peptide multiple alignment, which is very hard task for proline-rich peptide. Because, for example, if you have two peptide contain a class 1 motif, if you want to get a very good PWM, the peptide should be aligned in this kind of alignment. But if you use any automatic alignment program, you'll get this alignment, and which will give you a suboptimal PWM. And the other method is also published, for example, energy model approach. They are normally based on the solved 3D structure of protein complex structure. So, but in reality, the structure is known only in few cases. So here we developed a graph kernel support vector machine approach based on neighborhood subgraphs pairwise distance kernel, or NSPDK, developed by Costa et al. So here is our advantages. So our advantages are we do not need for an initial alignment of the peptide. And the models, our method models higher order correlation between the amino acid position. And it also integrates any additional type of information. So in our study, we use the physicochemical property of the amino acid. And our method is also efficient in training and testing and has linear complexity. So we developed two kinds of model. One is the single domain model and one is the multiple domain model. Single domain model is we developed a model for each SS3 domain. So we have studied 70 SS3 domain. For single domain model, we developed 70 SS3 domain, SS3 model. So how do we encode the features here? So you see here as a peptide sequence with a gray circle. We have a position as specific placeholder. That means we do not have any fixed position for a certain amino acid. And we introduce the physicochemical property, which is charge, hydrophobicity, and finally the amino acid type. Now, we also tried a different order of the encoding, and we found that this order of encoding gave us the very good discriminative model. Now, when we make a model for multiple domain, in that case, we not only consider the peptide information, but also consider the domain information. Only difference from these two graphs is in domain feature encoding, we have a fixed position for the domain because domains are easily alignable. So you have a domain you can align well, and you have a fixed position for the domain. And the white circle indicates the gaps present in the alignment. So each instance is represented as a direct disconnected graph into two components. One is for the peptide, and one is for projection of a given domain over the consensus alignment. So graph kernel is generalization of a k-mer kernel to directed graph, and each graph is a pair of small subgraphs, and each subgraph is a neighborhood graphs. So here, if you have the vertices, if you have there are two important parameters, which is radius and distance. If you have radius 1, from these vertices you have further 1 down, and you have when you have a radius 2, you have this kind of graph. When you have radius 3, you'll have this kind of graph. And when you introduce the distance from this is your root vertex. So here is the example of the radius 3 and distance 5. So you have a radius 3, you get this graph, kind of this graph here. And from here, you take the distance 5, which is, this is the vertex then. And then you take again the radius 3, and you get this one. Now we combine these two graphs and consider it as a feature. So that means using this, we are allowing the dependency among the amino acid position. So here, the graph encoding, here is given a graph, we decompose the graph into the graph features. So for given all combination of graph features, we count the frequency if the features are present in our graph encoding. For example, this one, if it is present here, we encode, we count the frequency. For if it is not present, we ignore it, and so on and so forth. We get all the frequency of the features that are present in our encoding. And then we used hashing technique to get the isomorphic identifier for each graph features. So it is an integer number, and it will give you the unique identifier of each features. And after that, we got the sparse vector from the isomorphic identifiers. And once you get the sparse vector, we use the SVM. Okay. So the time complexity. So our model is linear complexity in training and testing. So we can train one instance in 0.07 seconds. And this runtime includes the upload in main memory, generation of graph features, model in induction using stochastic gradient descent. And we can test 100,000 peptides in less than 2 hours in a normal desktop machine, which is also, the program is also trivially parallelizable. Now the data set, we retrieve all the data from PepSpot DB, which consists the chip experiment published by Carducci et al. Last year. So we found the interaction of 70 SST domain and almost 9,000 peptides. So in chip experiment, you would have, if you have a 70,000 domain and the 9,000 peptide, the possible interaction would be more than 650,000. So here in the pie chart, you can see all the combination of interaction. Now we only got the 16,000 positive interaction. So here the blue is indicating the 15,000 positive interaction, but one cannot assume that these all possible interaction are negative, because in high throughput technique the method is highly affected by the false negative data. So one cannot assume it. So here in the table you can see that around 3,000 of peptide involved in positive interaction. There are 16,000 positive interaction. Among them, around 500 are reported in MIN database, which is manually curated and highly reliable database. And negative peptide involved in negative interaction may be in all peptide, because one peptide can interact with one domain, but not interacting with other domain. So all are these are the unknown. We label that as unknown because we are not sure. And we retrieve high confidence negative. So I'll come later, how did we retrieve the high confidence negative? So now for the time being, you assume if we consider this blue is positive because it's a positive interaction, and rest of all that we labeled as unknown, we consider right now as a negative interaction. And then this is the result. The x-axis indicates the SST domains. We have 70 SST domain, and y-axis is area under precision recall curve. So the red one is our graph kernel approach with two class. So we consider the 16,000 positive here, and the brown area considered as negative. Even though the data is unbalanced, of course that is unbalanced because there is a lot of false negative data. So here, the blue one is our graph kernel approach with a single class. So we use the graph kernel approach only using the positive interaction. We ignore the negative interaction. And the MUSI is also based on the PWM approach. So that means it is based on only the positive class. So here you can see our graph kernel approach with one class and the MUSI is performed similarly. That means they are one class model. So the take-away message is the two-class model or discriminative model is better than the generative one, even though our negative is affected by the false negative. So now I'm going to talk about how did we filter the false negative data, or how did we get the highly confidence negative data. So given a interaction of a certain SS3 domain, for example, for one SS domain, you have a set of positive interaction. We start with positive interaction, and we group them according to their motif type that the SS domain can bind. So the canonical one and non-canonical one. And then we produce PWM for each motif type using meme. And we score all available peptides. In our case, there are 9,000 peptides, including the positive peptide here. So we score using the PWM and sort them according to E value. And we use the cutoff value for getting the high confidence negative. Now the cutoff value was chosen the highest E value of a positive interaction data. So now the gradient area, you can see these are the highly confidence negative data. We merge them together and take a non-redundant set of the negative. So now, when we use the filtered negative, the performance was jumped, and we got a very convincing AUCP PR curve. So the takeaway message is the performance improved by the negative filtering, and we reduced around 600,000 to 200,000 high confidence negative. So till now I have talked about the single domain model. Now I am going to talk about the multiple domain model. So the idea is to transfer the knowledge between the similar domains. If the domains are highly similar and very good alignable, so you can use one model for multiple domain. For example, we built a model for these 6 SS3 domain which are very high in sequence similarity. So that when we combine all the interaction data of 6 SS3 domain, we get 900 positive interaction. But if you use one single SS3 domain interaction data, you might have vary from 100 to 300 interactions. Now here you see the x-axis is the SS3 domains, the 6 SS3 domain, and y-axis is the AUCPR, and this is the 10-fold cross-validation result. So the red bar indicates the multiple domain model, the green one indicates the single domain model, and blue one indicate the MUSI performance. Now you can see the red one or the multiple domain model is always better than the single domain model because you allow to exchange the information among the domain. So the takeaway message is the better performance than the single domain model. So we did another experiment to identify the interaction of a novel domain. For example, if you do not have any prior knowledge of that novel domain and you would like to predict the interaction of that particular domain, you can use your multiple domain model if your novel domain is highly sequence similar with this domain. So here we take an example of LCK, which is also very sequence similar with this domain. And when we use the multiple domain, we got a AUCPR of 0.85, and if you use a single domain, you got 0.72. So here you can see that when we use this model, we have a lot of data point in our training set, the positive data point. So it's 900. But if you use the only one domain, you get a 150 positive data. So for the validation, we retrieve data from the MINT database, which is a manually curated and highly reliable database. And we got a 500 interaction, and we predicted 83% of them. So finally, we did a genome-wide analysis. We retrieve all the human protein, which are around 20,000 human protein. And we did a window analysis with the length 15 and step size 5. And we ended up with around 2 million of peptide. Here we introduced a filter approach, which is subcellular localization. Now, it's very unlikely that if domain containing protein and the peptide containing protein, they reside in different compartment of the cell, it's very unlikely that they would going to bind with each other. Therefore, we applied the filter here. So we allow only the domain containing protein and the peptide containing protein if they at least share one subcellular localization term annotated in Go database. Around we filtered 2,000 interaction data for each SS3 domain. And we report top 50 prediction based on the SBM score. And we also found that our prediction had biological insight because, for example, this protein, PI3C2B, interacting with many more SS3 domain predicted in our study. Actually, for validation, the two GRB2 and PLC1 already already validated that bind with this protein in the string database. To one step further, we also did a term-centric enrichment analysis using David tool. So, and we observed also the meaningful interaction. For example, P85 alpha bind with a potential group of protein that an important role in beta cell receptor signaling pathway, which is already known. So to conclude our study that we developed a alignment-free graph kernel approach to predict SS3 peptide interaction, which does not need any pre-alignment of the peptide, which also allowing the dependency among the position of the peptide, and also it's very fast and efficient and has a linear complexity. And moreover, the multiple domain model is better than the single domain model. If you have a domain which is very close to each other, you can use multiple domain model than the single domain model. The performance would be definitely good. So I'd like to thank my supervisor, Professor Dr. Rolf Backhofen, and I'd like to thank him for giving me the opportunity to work with him. And Dr. Fabrizio Costa, who is also a joint first author of this paper and also developer of NSPDK. And I'd like to thank BIOS and DFG for funding. And the models and genome interactions are available under this URL. We also apply the graph kernel method to RNA and DNA. So we developed a pipeline based on non-coding sequence structure to cluster 100,000 non-coding sequence in hours. And you can get it from this website as well. And thank you very much for your attention.
So there is plenty of time for questions. Question there?
So there are many structures of complexes between three domains and peptides. Did you consider using a B for refinement or better training?
So the solved structure is already, if you use the mean database, so we validate on the solved structure. So if you use the mean database, they are already manually curated and manually curated database. So we trained on the high throughput data, and we validate on the manually curated or solved structure. And we got like 83% correctly predict. Yeah.
Thank you for a nice talk. I kind of missed the logic behind the negative filtering? Could you just repeat just the basic assumption?
Yeah, sure.
Framework?
Sure. So in cheap experiment, if you have a 70 SS3 domain and you have a 9,000 peptide, so possible interaction would be more than 650,000 peptide interactions. Now, but one should not assume that these interaction that did not show in the chipsec as a positive interaction, these interaction is negative interaction, because these data is affected by lots of false negative data. So you have lots of false negative over there. So if you need to make a very good discriminative model, you have to have a very confidence negative data. So therefore, we filtered the confidence negative data out of these unknown data. So this was the motivation. So if you need to make the discriminative model, you have to have a very good, not only the positive data, but also the negative data. Yes.
So actually what you do is you have your known data, and then you say you build models for positive data, and then you build out the ones that are maybe positive by the models that you build. And then you get a high confidence.
Does that affect the evaluation, performance evaluation?
Yes.
Does that affect the performance evaluation?
Other factor?
So if you remove the gray ones during training, do you also remove them during performance evaluation?
Yeah, sure. Once we, so here we did not filter.
Oh, but, it's easier.
Yes.
Removing the hard ones.
Yes.
Okay.
But they're all very proline rich and they're all very
Yeah. So all the peptides studied in this chip experiment, all are proline rich. So there is no chance that you're taking the positive, the negative interaction data, which is too far from the positive. So then it would be very easy to predict the model. So these are close, but still are the Prolin rich.
Thank you.
Yeah, welcome.